All right, everybody, we're going to talk about the blood in chapter 14. Okay, so just a little intro. The average adult has about 5 liters of blood volume. 5 to 6 really depends on how big you are. Males tend to have more than females. A rule of thumb, about 5 liters, which accounts for 7% of body weight. So the 5 to 6 liters number you should remember as a norm. Don't worry about the body weight part. That's just a fun fact. All right, so if you remember back from our tissues chapter, blood is classified as a connective tissue. It's our only connective tissue that does not actually stay in a fixed location, which is very important because its primary function is to serve as a transport mechanism for the body. So kind of all of blood's functions kind of revolve around that concept of being a transport medium. So it delivers oxygen, it removes wastes, a big one of those being carbon dioxide. It delivers our nutrients, hormones, enzymes, pretty much any substance that our body needs to transport from one area to another. It helps protect our body from infection because we have white blood cells that are part of our immune system within our bloodstream. It helps stabilize the acid-base balance. And it also helps regulate our body temperature. So our blood vessels can constrict or tighten so that we get less blood to the surface to keep us warm, or they can dilate or open up and more blood will go to the surface, which helps us cool down. Okay, so talking about the components of blood. So if someone drew your blood, it would not look like this. This is what a blood sample would look like after it is spun through a tool known as a centrifuge. So for those of you who have not had a lab yet or have not used a centrifuge, it basically just spins it really, really fast. And oftentimes that will cause things to separate into its different components. And so the reason that we analyze these components when it comes to blood is that it can give us information regarding diseases and disorders in the body. And this helps us isolate different parts to be able to test them separately. So we're going to work from the top down here. So the plasma, this clear part, so that's your extracellular matrix. And that's about 55% of your blood volume. And we'll talk a little bit more about kind of what's in your plasma on the next slide. This little tiny white line is not space. That's actually known as the buffy coat. So that's your white blood cells and platelets. It's only about 1% of your blood volume. And then we have the formed elements. So this is where the actual cells and fragments of cells are contained. About 45% of blood. A majority of that is your red blood cells. But you can also, so you'll have definitely majority red blood cells there. You can still get a little bit of leukocytes or the white blood cells that are circulating. And you can also get platelets, which are really tiny pieces of a cell, really. And we'll discuss those in a few slides down the line here. Okay, so our plasma components themselves, it's mostly water. That's why it looks relatively clear. It also has some proteins. Albumin is the most important one to remember. It's just the most abundant. Our nutrients, electrolytes, hormones, gases, kind of all the water soluble stuff is going to be found in the plasma. So because of the proteins, it has a role in blood clotting. It carries some of our clotting factors with it as well. It can help immune function. And it also helps us regulate our fluid volume since there's a lot of water in it. Kind of changing the concentration of the plasma can regulate that fluid volume. So if you see the term serum, that is just plasma minus our clotting proteins. So not all the proteins, just the clotting ones. So serum is plasma without clotting proteins. We'll talk about the whole blood clot process down the line, so that may make a little more sense. Just right now, just remember serum is the plasma without clotting proteins. Okay, hematocrit is the percentage of RBCs in a sample, and because these are the heaviest, those formed elements go to the bottom. So remember we said there are some white blood cells, there are some platelets in those formed elements, but a large majority of that is red blood cells. Okay, So we'll talk about the actual formation of blood cells. So the process of the formation of blood cells is called hemopoiesis. So tissues that produce blood cells are known as homopoietic tissues. So we have our red bone marrow. That's kind of all our blood cells, red, white, platelets, anything can come from the bone marrow. And so that's in the ends of our long bones. You remember that's in the bone chapter, and as well as our flat irregular bones, the sternum, the cranium, stuff like that. Our lymphatic tissue, which is a couple chapters down the line, is the lymph system, produces just lymphocytes. And so keep that in the back of your mind right now. They play a role in the immune system. So they're found in the spleen, our lymph nodes, and the thymus gland, if you remember that from the endocrine chapter, last chapter. So we'll circle back for lymphocytes. And all blood cells begin as a stem cell. I'm sure a lot of you have heard that term in terms of the controversy behind some of the research that goes into to stem cells. But basically, a stem cell is an unspecialized cell. So it could really turn into a red blood cell, a white blood cell, or a platelet. It can really turn into any cell, but we're talking about it specific to the blood system. Excuse me. Okay, so this slide has a little more info on it than you actually need to know. So as we said, our blood cells start as the stem cell. That's our unspecialized cell. So it's going to give rise to these immature cells initially. And you guys do not need to memorize this whole chart by any means. It's just kind of giving you an idea of how our cells develop. So from the stem cell, depending on what type of cell it's going to then develop into, it kind of specializes there. And so then as these mature and divide, we get a second level for a lot of these. And then this is really what we're going to focus on, is this bottom portion here. So red blood cells are also known as erythrocytes. And so the process of creating these is known as erythropoiesis. So blood cells in general, that's that hemopoiesis. But erythropoiesis, if you see that term, it is discussing red blood cells specifically. There's a variety of different white blood cells, and we'll talk about the classifications down here as we go further in. And then thrombocytes, these are your platelets. That's what I was saying is really pieces of this larger megakaryocyte cell. We'll go through kind of each section individually. Okay, red blood cells, super important because they deliver oxygen to the cells. And so they're always represented by these little discs because that's how they actually look. And the form or their shape is actually important to their function. So it gives a large surface area, which helps aid with the diffusion of oxygen. So if you think our blood cells are moving pretty quick through a lot of areas in our body, so they have to be really efficient at dropping off and picking up what it needs to. So that large surface area allows it to do that. It's also considered biconcave. So you have this little tiny indent, and that's on both sides. And that helps them kind of be flexible and bendable. Our capillaries and some of our smaller vessels are super pressurized and super tiny. So for our blood cells, they have to be able to squeeze through those capillaries without breaking apart or getting stuck. So that their shape also allows them to stay flexible. So of course they have to remove carbon dioxide. That's a big part. They cannot replicate. So our red blood cells, once they're a red blood cell, that's it. They don't divide further down the line. So they don't have a nucleus and they don't have DNA. So that kind of takes replication out of the equation. And as we said, they have that flexible cytoskeleton to help them fit through our narrow vessels. All right, so this molecule here is really a zoom in into our red blood cell. This is known as hemoglobin. So this is a really relatively large molecule. It takes up 1/3 of our red blood cells interior. And so kind of has 4 different proteins that all come together. And these 4 protein balls are known as globins. And then each green guy here, that's known as a heme. And so that is an iron-based molecule that combines with the globin. The reason those hemes are important is because oxygen can bind with it. So the globins plus the heme creates a hemoglobin. Once the oxygen binds onto it, it is now an oxyhemoglobin. So each heme molecule can combine with one oxygen. So each hemoglobin can transport 4 oxygen molecules. And that is definitely important to know. One red blood cell can transport 4 oxygen molecules. Okay, first review question here. What is the extracellular matrix of blood? A, hemoglobin, B, red blood cells, C, plasma, and D, serum. Okay, the answer there is C, plasma. If anything, you should have had it narrowed down between C and D. Serum is plasma with the clotting proteins removed, so that wasn't quite specific enough to be the answer for serum. That's why we went with plasma. Okay, so as I said, erythropoiesis is the actual kind of creation of red blood cells. And as I said, they can't divide at all, so they kind of have a lifespan to them. So basically, what happens in our body is oxygen levels start dropping. This is because old red blood cells are being removed from circulation. So they can last for about 120 days. And then they go to our spleen where they are broken down and kind of eaten by some of our other cells to recycle the parts. So as these old red blood cells are removed from our circulation, we get a drop in oxygen levels because we have less red blood cells to carry our oxygen. So our kidneys have receptors to detect that change in the oxygen level, and they're going to secrete erythropoietin. So that an abbreviation for that is EPO. So that erythropoietin, it stimulates our red bone marrow to create erythrocytes. Remember, that's our other name for red blood cells. Reticulocytes are released. Those are your immature red blood cells, and they're going to mature into red blood cells. As we get more mature red blood cells, our oxygen levels go back up. Our erythropoietin and our red blood cell production goes back down. So this kind of is constantly working all the time. This stays constantly in balance. It's not like we make a bunch of red blood cells and 120 days later they all die. We have to make them all again. So this gradually happens. So it's not like we're dropping oxygen levels to a point where it's noticeable or dangerous by any means. Our kidneys are pretty sensitive to any changes, and so they continue to kind of cycle in new red blood cells as some of the old ones are taken away. And this is just a little animation that kind of walks you through these 5 steps. Okay, so as I said, we don't just get rid of the red blood cells completely. We really are trying to recycle them. So macrophages in your liver and spleen, that's the type of cell, will ingest and destroy the old RBCs. And it's really like recycling because we're going to take the hemoglobin and break it down back to its component parts. So remember the 4 globins and the 4 hemes. So that globin is a protein. So we're going to break that down into amino acids. If you remember from back when we talked about proteins and carbs and fats and that kind of stuff, we can use amino acids for energy, but typically we really create new proteins with those. And then the heme is broken down into iron and bilirubin. So bilirubin is probably a new term for most of you guys. We'll get to that down in the urinary and digestive system chapter. So the iron itself goes to the bone marrow to help make new hemoglobins. And then the bilirubin is excreted through the intestines as a component of bile. So you should know that those are the byproducts, but you don't have to really know anything besides that. Okay, moving on to white blood cells. Another term for these are leukocytes. This is the fewest of our formed elements, but super important because this is our immune system. They protect the body against pathogens. So all leukocytes actually do contain a nucleus. And we have two major classifications, granulocytes and agranulocytes. So I'm going to sneak back up to this picture here. So granulocytes, you see here, see all the little dots? Those are your granules. And so that's why they call them granulocytes. And then under the microscope, when they dye these guys, the different granules pick up colors differently. For example, I think basophils typically show up as blue. Eosinophils, they tend to dye red, and neutrophils, they tend to dye purple. So that's how they can differentiate when looking at it in a microscope. And then you have your agranulocytes. That's your lymphocytes and your monocytes. So you see they really don't have any of those granules in the cytoplasm. Okay. So these are a little different than the red blood cells themselves. Our red blood cells kind of keep circulating and circulating and circulating. So we're going to talk about granulocytes first. And so these all circulate for about 5 to 8 hours in the actual bloodstream. And then after that, they actually will migrate into a tissue somewhere in the body. So they all go different areas. And they can survive another 4 to 5 days there. So when they're in circulation, they're doing their job as an immune system cell. And when they're in the tissues, they're serving that same function. Okay, so neutrophils are the most abundant by far. That's 60 to 70% of all the white blood cells in circulation. So they're super mobile, and they kind of pop in and out of the blood vessels to spaces to engulf our foreign material or stuff that should not be there. So remember that's known as phagocytosis, when a cell kind of eats another cell or debris. The eosinophils are only about 2 to 5% of our white blood cells. And we find these most concentrated in the digestive and respiratory tract. They're involved in allergic reactions and kill parasites. And then we have our basophils. This is the fewest of the circulating white blood cells. They don't really have the ability to perform phagocytosis like the neutrophils, but they do have a very important job. They secrete heparin, and so that prevents clotting. So that white blood cells can actually enter an area in need. A lot of times when we get a cut or something like that, there's a lot of small bacteria and microorganisms that we really do not want into our body. And if the blood clots so fast that our white blood cells can't get there, that it kind of traps them in and our white blood cells can't get rid of them. The second thing basophils do is secrete histamine, which is actually kind of like a chemical signal to white blood cells that, hey, we need you guys over here. All right, the agranulocytes, we have our lymphocytes. So this is our second most numerous, 25 to 33% of white blood cells. It's very small, but it does a lot of important work for our long-term immunity. So this is the major worker of the lymph system, and it's a huge player in our immune system. So we have both T and B lymphocytes. So our T lymphocytes are kind of our attack cells, our fighter jets. They're going to directly attack the infected cell, cancerous cell, anything that we need to get out of there. The B lymphocytes are the memory aspect. So they produce antibodies against a specific antigen. So antibodies are what our body uses to identify something that's not supposed to be there. And an antigen is the foreign material or whatever is not supposed to be there. So the first time we encounter an antigen, we have to kind of identify it prior to deciding whether to attack or not. The second time that antigen is introduced, our B lymphocytes already remember that it's not supposed to be there. So we have antibodies which signal the T lymphocytes to attack it right away. So that's why it's good for long-term immunity, because we have our attack cells and our memory cells. So last but not least, we have monocytes. So these are about 3 to 8% of the white blood cells. It's our largest one, very phagocytic. So its job is to engulf really large bacteria and viral infected cells. So um, they're big because they have to eat big things. Um, they circulate for 10 to 20 hours, and then these also will migrate into the tissue and they transform into macrophages. And that's a big worker in our body in general. Remember, the macrophages eat the red blood cells. We talked about that earlier. And these can actually live for a few years. And so they function very similar to how a monocyte functions in circulation, but they do it in our tissues. So just to clarify, you guys do not need to know the actual percentages themselves. You should definitely know that neutrophils are the most abundant. Make sure that you pay attention to what each one is involved in the most, or kind of its role in the system. But the smallest, largest, like that kind of stuff, you don't need to remember. Definitely know neutrophils are the most abundant. Kind of pay attention to which ones are the granulocytes, agranulocytes, and know their functions, but don't get caught up in the tiny details. All right, our last category is platelets. These are also known as thrombocytes. So second abundant, most abundant of the formed elements, even though they're very tiny themselves. And so they're really there to help us stop bleeding. Another kind of medical term for bleeding is hemostasis. And as we said earlier, our platelets are really fragments of this megakaryocyte cell, which is found in our bone marrow. Sorry, guys, I'm playing a little bit of a cough. Okay, this you do not need to study or memorize by any means, but this is just an actual blood smear, or what a sample of blood would look like under a microscope. So you see we have a little tiny platelet there. You can see our eosinophil, all the granules in there. Our lymphocyte, no granules, just stained. Our neutrophil, they're a lot tinier, but they're there. And then all these guys are our red blood cells, so they're by far the most numerous. This is just another example with some of the stains. So these stains aren't super contrasting, but if you see the colors are picked up just a little bit differently, and you can still see granules and not. This one's a little easier to tell. Okay, second review question. A move to high altitude would trigger which change in the blood? All right, think about it and try to decide on an answer, and I'll give you a hint before I actually show you. So think at higher altitudes, our blood is not quite as oxygen rich, or it's harder for our oxygen to diffuse into tissues. So think if that would change your mind at all. Okay, so the correct answer is A, which are increased reticulocytes. Remember, the reticulocytes are immature red blood cells. And so when we have lower levels of atmospheric oxygen, such as areas of high altitude, we then need to increase our production of red blood cells so that we have more carriers to get that oxygen somewhere more quickly. So we need more reticulocytes to mature into red blood cells. The other one in here was neutrophils. Those are white blood cells. They don't really respond to that change in altitude because they don't really have the oxygen influence. Now, if we got sick, then we might get some more white blood cells compared to normal. Oftentimes, elevated white blood cell count is one of the first signs of an infection or another issue in the body, but not going to be affected by altitude or oxygen levels. Okay, we're going to talk about bleeding now, or really how to stop bleeding, or how our body stops bleeding. So the first step is what's known as a vascular spasm. So our vessels have smooth muscle fibers that line the walls. And so when we get damage, those actually contract and help restrict how much blood is flowing through. So they make it a smaller area so that less blood comes flying through. It's a very temporary response. It's kind of just to literally slow the bleed before we can get some more help. All right, step 2 is what's known as the formation of a platelet plug. So as we said, our thrombocytes or our platelets are really, their main job is to stop bleeding. So that broken blood vessel will expose these collagen fibers that are kind of on the wall of the vessel, and this creates a little bit of a rough spot for our platelets to stick to. So it's kind of a silly term, but when our platelets stick to the collagen, they're now known as sticky platelets. And so these sticky platelets stick to the vessel wall, and then they stick to each other, which ends up forming that platelet plug here. So chemokines are kind of a type of chemical signal that our body can utilize, and they actually facilitate platelet clumping. So when the chemokines get to the area, we get further constriction. The vessel shrinks, and it kind of calls more platelets to the area. So that temporarily seals the vessel wall. Our platelets are tiny and they, like I said, they're sticking together, but they're not super strong. So that, again, this is kind of a temporary hold before we can really have a permanent solution. Okay, so step 3 is the formation of our actual blood clot. Blood clotting is also known as coagulation, and it really is kind of a sequence of chemical reactions that all rely on proteins known as clotting factors. So the clotting kind of cascade, which we're not going to go super in depth into, is kind of like a domino effect. So kind of each protein activates or attracts the next one. And so there's 2 different ways that our body can enter that kind of clot cascade. And so it really depends on where the damage came from. So the extrinsic pathway really means outside of the body, or outside of the blood will release our clotting factors, whereas the intrinsic pathway is triggered when clotting factors are activated within the blood, such as that platelet one. So I don't think we're going to go through this intrinsic and extrinsic pathway, but I do not necessarily need you guys to remember every detail of this diagram. When I put out your study guide like normal, I'll try to simplify it for you, but let's just walk through it together. So here we have remember our collagen threads that get our sticky platelets coming in. So factor X is really our most important clotting factor or clotting protein. So that's the end result of both our extrinsic and intrinsic pathways, but the way we get there is a little bit different. And so once we get to factor X, we're going to get a blood clot. But the extrinsic and intrinsic pathways have different ways of getting to factor X. So you guys should definitely know that factor X is kind of the last step prior to the blood clot, or that's what really triggers our clot to form. So once we get to that factor X, the end result, our body is going to produce an enzyme called prothrombin activator. So we're ignoring the difference between intrinsic and extrinsic. We're at the end of the pathway. We're at factor X. Prothrombin activator is then released. That prothrombin activator acts on globulin called prothrombin. That's another protein. That prothrombin is then going to be converted into thrombin by prothrombin. This thrombin is then going to transform a plasma protein known as fibrinogen into fine threads known as fibrin. So let's walk through that again. So factor X is the end of the extrinsic and intrinsic pathways. We then get prothrombin activator. Prothrombin activator acts on prothrombin to convert it to thrombin. Thrombin then transforms fibrinogen, which is already in the plasma, into fibrin, which cannot dissolve. So those fibrin threads basically kind of form like a web around that platelet, kind of our sticky platelets. So we then get more platelets, more red blood cells actually going through the web and kind of getting caught. And that's what actually creates the clot. So our clots are a big bunch of fibrin threads, blood cells, platelets, kind of all stuck together. So in a really small vessel, this clot here can actually do a really, really good job of plugging up the hole completely. But oftentimes, if it's a large enough insult or injury, or if it's a super large vessel, even our clotting factors or our pathways are not enough to actually stop the bleed. But in small vessels, this works great. Okay, so we want the clot to last as long as we need to, but once we heal, we don't necessarily need that extra junk clogging up our vessel. So like I said, the fibrin is trapping all the platelets and red blood cells, and so that actually pulls the walls of the vessel a little bit closer together. And so as that heals and kind of gets pulled in closer together, we eventually get our cell or our vessel wall back. And so we then get another domino reaction, which results in our turning plasminogen into plasmin. So plasminogen is just floating around. When we need to get rid of our clot, our body converts plasminogen into plasmin. Plasmin dissolves the fibrin meshwork, which breaks up that clot. So fibrins are spider web. Plasmin gets rid of fibrin. Okay, and then to actually prevent blood clots is also important, because when we don't need a blood clot or if we don't have an injury, blood clots can be very, very problematic. So that smooth endothelium that lines our vessel walls is super smooth so that the platelets do not stick. Remember, when it breaks and we get that rough surface from the exposed collagen, the platelets are attracted to it. The blood flow itself helps prevent blood clots. So our continued circulation dilutes thrombin, which is what activates that fibrin. So even though the thrombin is there, it's not actually kind of concentrated enough to trigger a clot. And then if we're not getting that normal blood flow, such as if we sit for a really long time, thrombin can actually accumulate and lead to a clot formation. Oftentimes, after people get a surgery and are on bed rest or kind of laid up in the hospital for a while, one of the biggest concerns is blood clots, because our movement and kind of just our daily lives keep our blood flowing. And when we stay still, that blood starts to pool and doesn't flow nearly as kind of frequently and consistently. And so if thrombin actually is able to accumulate, a clot can form. And then anticoagulants. So basophils and mast cells secrete heparin, which you talked about earlier, and that actually blocks the action of thrombin and prevents it from happening. So we kind of always have a maintenance level of heparin being secreted to prevent blood clots as well. Okay, third review question here. What is the first step in hemostasis? So this is the first step in stopping the bleed. A, formation of a platelet plug. B, formation of fibrin. C, formation of a thrombus. Or D, vascular spasm. And the answer is D, vascular spasm. So that's our very first step. We then get our platelet plug, and then we actually form our blood clot. A thrombus is a good term to know or be aware of. That's basically an unwanted blood clot that's in a blood vessel. So thrombus is another term for a clot, but only when we do not want it there. Okay, we're going to talk about blood types next. So I'm sure you all have heard of the different blood types, but I'm not sure if you know why they are known by the different letters. So each blood cell has an antigen, which is also in this case known as an agglutinogen. So when it comes to our blood, we have antigen A and antigen B. So A is represented by these green triangles, B is these kind of orange dots, and AB has both the A antigen and the B antigen, and type O has none. Okay, so our blood cells have the antigens, while our blood flow, or the blood itself, contains our antibodies or agglutinins. So we talked about the antigens and antibodies when we talked about lymphocytes. So same concept here, but the antigen is on the red blood cell or not, and the antibodies are present in the blood. So, for example, blood type A, the red blood cells have an A antigen, and their antibody is an anti-B antibody. So they're fine with the A's, but they would not be happy with B's. Vice versa, blood type B has the B antigen, but the anti-A antibody. So they're happy with B's, would not be happy with A's. Which is why you see no antibodies in blood type AB, because either anti-B or anti-A would attack the red blood cell. And then blood type O is the opposite, because it doesn't have any antigens on its surface. You see both anti-A and anti-B antibodies. So if someone with blood type B was given blood type A, it would be the antibodies would attack that new blood as a foreign substance because of the recognition of the wrong antigen. So blood type AB, they can receive blood from anybody because they don't have any antibodies to fight it. So if you're blood type AB, you're pretty safe. Blood type O is loved by the blood banks because it's a universal donor. It doesn't have any antibodies, so we can give that, or any antigens on its surface, so we can give that to anybody, A, B, AD, or O. Now, you guys might be thinking I'm leaving something out in terms of the positive and negative that you hear. People say I'm A positive, B negative. So that part actually refers to our RH factor. So I'm going to circle back here for just a second. This is just that example that we talked through. That's that type A antigen, type A blood, A antigen, anti-B antibody. If we introduce type B blood with the B antigen, our anti-B antibodies are going to attack and basically will clump together. Okay, so this is where the positive and negative comes in. So this is known as the RH group. So if someone says they're A positive, that means they're blood type A, and they also have the RH antigen. RH negative does not have that RH antigen. So a large majority of people are RH positive. And so there's really two ways that you can actually develop anti-RH antibodies. So antibodies that will attack the RH antigen. So if you're already Rh negative, but you get Rh positive blood, your body is going to create anti-Rh antibodies because it's not used to that antigen being present. The other way this can happen is if an Rh negative person or woman is pregnant and their baby is Rh positive, because the mom and the baby are going to share blood flow. So that Rh positive blood is going to be introduced into the woman's system as well. So they'll create anti-Rh antibodies. So we'll talk about why this is actually important here in a second. Okay. So this is someone who has negative. They are Rh negative. So they get an infusion of blood that has the Rh antigen, Rh positive blood. In response to that, they create anti-Rh antibodies. So no big deal the first time, because if the first time was introduced, the antibodies aren't there yet. It's just that first introduction of positive blood creates or causes the body to create those antibodies. So then the next time they receive Rh positive blood, those antibodies are already going to be ready to attack that Rh group, and that's going to cause the red blood cells to agglutinate or clump. The same concept with pregnancy. You have an Rh negative mother and Rh positive baby. The mother is exposed to that Rh positive blood. So you know what comes next. That means we're going to create antibodies, just like the transfusion. So then next pregnancy, if they have an Rh positive baby again, those antibodies are going to attack the baby. No good. They do have, I believe it's an injection that they give now. Called RhoGAM. It's in your book. This is actually on your fun fact underneath these images in your book. So it's not like this is end of the world type thing. They have preventative measures for this. It's just understanding kind of how this concept works. So in both cases, it's not really the first time that the posit is introduced that there's a problem. It's the second time. Okay, I think this is our final review question here. What substance carried by each red blood cell determines blood type? Is it A, antibody, B, antigen, C, hemoglobin, or D, globin? So you should be able to narrow this down to 2 at least. And the answer is B, antigen. So we're talking about the red blood cell itself. Remember the type A was that one with the green spikes. Type B was the little orange dots. So those are the antigens. Our blood plasma has antibodies against either A has anti-B or B has anti-A. And then hemoglobin, that gives our blood cells its red color and can carry some oxygen. And the globin is just a half of that equation there. All right, so that is the blood. And next we will talk about the heart, and then we'll talk about kind of the whole vascular system together, and that'll wrap up our next test.